Yes, we can hear you.
Okay, great.
All right, so Mackenzie Fitzpatrick is a graduate student in Leah Solberg's lab working on HS rats and diabetes, etc. So she'll continue this theme into the next talk, and we're ready whenever you are, Mackenzie.
Okay, great. Thank you. So I'm Mackenzie Fitzpatrick. I'm a grad student in Leah Solberg Woods' lab at Wake Forest. And the title of my talk for today is A Transmembrane Domain Mutation in ADCY3 Causes Obesity in Rats via Altered food intake or energy expenditure depending on sex. So obesity is a growing epidemic in the US and globally, and the prevalence of obesity in the US is expected to reach 50% by the year 2030. Obesity is a complex trait, and most cases of obesity are polygenic, so multiple different genes contribute to it in addition to the environment. I know that with this audience, I don't have to spend too much time on the HS rat, but our lab uses HS rats to study obesity. They're an outbred rat model that originate from 8 inbred founder strains, and they're a great tool for fine genetic mapping of complex traits, including obesity. Back in 2018, our lab performed a genome-wide association study in HS rats, and we found a QTL that was associated with adiposity. This QTL contained a protein coding variant in a gene called ADCY3. So the leucine at position 121 was changed to a proline. And this variant was predicted to cause a bend in the transmembrane helix of the ADCY3 protein. So you can see L121P tagged in red in the rat sequence over here. And this variant that's tagged in yellow is a variant that was identified in humans in association with childhood BMI. And this variant is in the same transmembrane region as the variant that we found in rats. The protein encoded by ADCY3 catalyzes the synthesis of cyclic AMP. ADCY3 is a membrane-associated protein. It's expressed broadly throughout the body in both humans and rodents, but primarily it's expressed in the brain, in the hypothalamus, in the olfactory regions, and it's also expressed in adipose tissue. And some of the downstream effects of this cyclic AMP pathway include thermogenesis, adipogenesis, and also energy homeostasis and satiety signaling. In the human literature, several different variants in ADCY3 have been identified in association with obesity. There's a bunch of information in this table, but what I really want to point out is that some of these variants that have been identified were causing decreased ADCY3 expression in association with obesity. But there have also been several protein coding variants in this gene that have been found. So this is the one I was just talking about 2 slides ago. And several others have been found as well in the catalytic domains, the transmembrane domains. So both expression altering and protein coding variants have been found in this gene in humans. Also in the literature, several ADCY3 mouse knockout models have been done in the past, and they've been found to have similar phenotypes. So obesity and larger fat pads, increased leptin, increases in depression-like and anxiety-like behaviors, changes to glucose tolerance, decreased physical activity, and increased food intake. But there are still several outstanding questions based on the mouse knockout studies that have been done. The first problem is that these rodent studies that have been conducted so far have only used knockout models. These knockout models have limited translational relevance to humans, especially given that among the human variants that have been found, most of those either only partially decrease the ADCY3 expression, and some of them don't affect the expression at all and are instead protein coding variants. The other outstanding question is, what is the cause of this adiposity in these knockout models? So some of the knockout studies have found changes in food intake. Some of the studies have not found changes in food intake. And there actually has not been a study yet that has looked at energy expenditure in an ADCY3 knockout model. So to start to address some of these questions, our lab is specifically asking if a mutation in the transmembrane domain of ADCY3 can produce the same phenotypes that have been seen in the knockout models. And specifically, what is the cause of the adiposity in this mutant strain? So to answer these questions, we and some colleagues at MCW generated a rat model that has an ADCY3 mutation in the transmembrane region. So this mutant strain has a 3 base pair deletion, and that results in a change in 2 amino acids in the protein. So it results in the loss of a phenylalanine, and also the valine here is changed to a leucine. The first thing that we wanted to confirm is that this mutation doesn't actually alter the expression of ADCY3 to demonstrate that it is not just another knockout model. So when we looked at the RNA and the protein expression of ADCY3 in the fat pads of these rats, we didn't see any differences in expression, and we looked in both the males and the females. So this is confirming that we really have a protein coding variant model rather than an expression altering model. We then conducted an in vivo study with these mutant rats. So we used males and females, wild type and mutant animals. They start on a high fat diet at 5 weeks of age. They're on the diet for several weeks, and then they get an echo MRI to measure fat mass and lean mass. They have an intraperitoneal glucose tolerance test to look at glucose and insulin levels. A few weeks later, they go into the TSE Phenomaster chambers. And the TSE chambers are indirect calorimetry chambers. So they measure oxygen consumption, carbon dioxide output to get a measure of energy expenditure. And they also measure things like food and water intake, locomotor activity, etc. And finally, we euthanize the animals and collect tissues. And throughout the course of the study, we collect body weights and cage-wide food intakes for all of the animals on a weekly basis. We found that the mutant rats do weigh more than the wild type rats over the course of the study. So in the body weight curves here, we can see that for the mutant males and also for the mutant females, they do weigh significantly more than the wild type animals. When we look at their body composition, we can see that this body weight gain is entirely due to a gain in fat mass with no changes to their lean mass, again holding true for both the males and the females. So this really tells us that we have an adiposity phenotype rather than the animals just being larger or weighing more. The next two slides are looking at our data from the TSE Phenomaster chambers. So this is showing a snapshot of 24 hours from when they're in the chambers. And the shading on the graphs, so the white shading represents their light cycle when the lights are on, and the gray shading represents the dark cycle. Ignore this little bar of gray at the end here. That is unfortunately a bug in the analysis program that we were using that has not been fixed yet. This should be white all the way down to the end. So when we look at the male data, we can see that the mutant males consume significantly more food than the wild type males over the course of 24 hours. But when we look at the energy expenditure data, we didn't see any differences in energy expenditure in the mutants. One thing that's important to know about the analysis that was done here, so we used a program called CalR. It's a great program for looking at indirect calorimetry data and starting to make sense of it. And one of the things that CalR does in the statistical analysis is that it accounts for the effect of body weight on several of these phenotypes with an ANCOVA. So because the mutant animals are larger than the wild type animals, we would expect them to have a higher energy expenditure just at a baseline. So even though there's a visual difference here between the animals with the mutants expending more energy, that doesn't come out to a statistically significant difference when we account for the effect of body weight on energy expenditure. And it's the same story here for cumulative energy expenditure over that same time period. When we look at the females, we see the opposite of what we saw in the males. So there were no differences in food intake between the wild type and mutant females, but the mutant females actually expend significantly less energy than the wild type females over 24 hours. And we see the same here when we look at the cumulative energy expenditure, a nearly significant trend in the same direction. So these results are telling us that the adiposity phenotype in the males is driven by changes in food intake, and in the females, it's driven by changes in energy expenditure. And interestingly, this is not the first time that we've seen sex differences in this mutant model. So the first sex differences that we saw when our colleagues at MCW generated this rat model for us, they gathered some preliminary body weight data in the animals on a chow diet. And on a chow diet, they found that the males had a difference in body weight, but not the females. This adiposity sex difference on a chow diet is actually also reflected in the ADCY3 knockout literature. And this is one of the main reasons that all of our rats for this study are on a high-fat diet. We also found sex differences in serum insulin after an overnight fast. So we found that the mutant males, but not the mutant females, had significantly increased serum insulin. And we also see sex differences in a series of cold exposure studies that we're conducting. So for these studies, we put the rats into a cold room and measure their body temperature as a way of measuring thermogenesis, which is also tied to energy expenditure. I should point out that these graphs here are from an ADCY3 knockout model that we also conducted in rats. I don't have the data yet for the mutant strain, but it is coming. But at least in the knockout rats, we see that for the males, there's no difference in how the wild type and the knockout males maintain their body temperature. But for the females, we can see that the wild type females are able to recover their body temperature much better than those knockout females. So this agrees with what we've seen in the TSE chambers in terms of energy expenditure differences in the females. And it is another example of where we see sex differences in this mutant model. So in conclusion, we found that rats that have a transmembrane domain mutation in ADCY3 show adiposity that is similar to the ADCY3 knockout models that have been done. And these results really underscore the importance of the transmembrane domain for protein function. It's interesting that we saw a similar adiposity phenotype in our mutant model compared to the knockout models, despite the fact that they have different underlying genetic causes. And we also found that the underlying cause of the adiposity depends on sex. And these results really underscore the importance of looking at both sexes in genetic obesity models. This is especially relevant given that sex differences have been reported in the literature in obesity-associated genetic variants in humans. We still have several things left that we want to do with this mutant rat model. So the first thing is to look at some of the molecular mechanisms underlying this mutation. We primarily want to look at how it's affecting protein localization and also protein function to cause the phenotypes that we're seeing. We're planning to dig a little bit more into the sex-specific differences that we've seen, specifically looking in both the brain and in adipose tissue. We're hoping that once we get some more insight into the molecular mechanisms, it'll give us further insight into why we're seeing these sex differences in specific phenotypes. And finally, the last thing that we're planning to do, I didn't talk about this today, but ADCY3 has also been associated with depression in humans, and it's been proposed as a potential blood biomarker for diagnosing major depressive disorder. So we're conducting an arm of this study where we are looking at behavioral differences in the mutant animals and trying to see if we can see a behavior phenotype and an adiposity phenotype in the same model. So I'd like to thank everybody in the Solberg Woods Lab who helped with this project, the ARP staff who care for our animals, our colleagues at MCW, several of our other collaborators, and my thesis committee, and the funding sources. And I would be happy to take any questions.